Biological applications of micro electromechanical system and nano electromechanical system. Okay. Uh, Now, in this class today, we are going to discuss microfluidics. Microfluidics. Okay, for solution control and input-output architecture for bioreaction readout. So, this microfluidics is used for solution control. There are a lot of applications for microfluidics in the by using MEMS and NEMS. Okay, so actually we are calling the microfluidics as a cornerstone of MEMS and NEMS technology. Okay, microfluidics is a cornerstone of MEMS and NEMS technology. Why we are calling it is a cornerstone? Because which enable precise control of tiny fluid volume for various biological and chemical applications. So we are it is a we can call it is a cornerstone of MEMS and NEMS technology. So by integrating this microfluidics, we can do various advanced researches. Okay, we can develop various high efficiency system for managing and monitoring bioreactions by using microfluidics. So they have a lot of applications like diagnostics, drug discovery and synthetic biology, etc. So we are going to understand things deep. So this picture, which is a AI generated image, which is represent biological applications of micro electromechanical system and nano electromechanical system. So this figure contains three parts. First one, microfluidics. You can see this part, this curved path. Okay, this curved path represent that is the fluid coming into the reaction chamber. This part is called the reaction chamber. This part is the reaction chamber, and this is the path channel by which fluid is coming into the reaction chamber. So this is represented as a microfluidics. In this side also this path is representing microfluidics. So this channel which enable precise control of tiny liquid volumes. Okay, so this fluid coming into the reaction chamber. The second part is reaction chamber, that is we can see this part is the reaction chamber. So this have pink circular cells and orange dot. Okay, that is we can the components of the reaction, that is nutrients or reactants, etc. So where bioreactions is like enzyme activity or cell growth or DNA amplification. Okay, this type of bioreactions may occur in the reaction chamber. So this part is the microfluidics part and this part is the reaction chamber. There is a third part here, input-output architecture. So which contain nodes and connectors. So these symbols electrical or optical readout system. So we can measure the parameters of the reaction chamber by using this input-output architecture. So these three parts, first one microfluidics, second one reaction chamber, third one input-output architecture. So these three parts components are part of the system. Okay, so this is the how microfluidics and the input-output architecture are integrated for biological process. So in this case, in this diagram, you can see that microfluidics and the input-output architecture were integrated for biological purpose, or we can say it is a biological process. In this class, you are going to understand the role of microfluidics in NEMS or MEMS, that is role of solution movement and control, and microfabrication for microfluids, Then various applications in bioreaction management, that is point of care diagnostics, drug discovery and screening, then synthetic biology, environmental and food testing, advanced research applications. First we will understand the role of solution movement and control. So first point, that is a miniaturization. So this microfluid, by using this we can handle nanoliter or picoliter range. Okay, we can miniaturization, it is possible. The second is we can control precision flow control. Okay, that is with high spatial and temporal accuracy by using various micro valve, pump, channel. Okay, so we can handle with high spatial and temporal accuracy. Third one, we can handle fluid containing DNA, protein, biomolecules, okay, etc. So these are the role in solution movement and control. There is a miniaturization. In that part, we can say that we can control nanoliter volume or picoliter range. The second thing, precision flow control. In that part, we can say high spatial and temporal accuracy. And the third one, we can handle the fluid containing cells, DNA, protein, etc. Then microfabrication for microfluidics. So this diagram, it is a AI generated picture. It is a complex geometry. It is an example for MEMS NEMS technique. Okay, by using lithography or etching, so which can create microfluidic structure with a complex geometry by using polymer, silicon, glass, etc. Okay. Then point of care applications in bioreaction management. Okay, point of care diagnostic. We are familiar with the various blood tests for many diseases, infection diseases. So during the lab test, blood test, we will combine microfluidics and readout system. So we can perform multiple diagnostic tests on a single platform, that is lab on a chip devices. In these devices, we will combine microfluidics and readout system to perform multiple diagnostic tests on a single platform. There will be on a single platform we can do multiple tests by using lab on a chip devices, by using MEMS or NEMS. Similarly, we can do rapid detection. Okay, that is an automated microfluidic system which reduce the time. So we can do the test rapidly. That is another point of care diagnostics. Then drug discovery and screen. This microfluidic channels have advantages applications in biological application, that is high throughput screening. In this case, you can see that these channels. Okay. So this high throughput screening, which means that there is a parallel microfluidic channel, 1, 2, 3, 4. Okay, parallel microfluidic channels. So these channels enable testing of multiple drug candidates simultaneously. We can do multiple tests simultaneously. That is the application of this one. Then reaction optimization. Then in this case we can control. You can see here there is a valve here, there is a pump here. Okay, it's a microfluidic. So we can tune control. We can fine tune the control of reagent delivery. So we can enhance the reaction condition for drug discovery. So we can say that fine-tuned control of reagent by using this technique. In this, we can control the motion. Okay, that is the reaction optimization. We can optimize the reaction by tuning various parameters, flow control. In this case, we understand that there is a parallel channels, so we can enable testing of multiple drug simultaneously. In this case, you can see that there is a fine tuning. That is, we can say it is a reaction optimization. Then these have a lot of application in synthetic biology, that is gene and protein synthesis. It is just an AI picture, AI generated image for gene and protein synthesis. So we can say this microfluidics support precise control of reactions. Then cell-free system. So there will be automated microfluidic platform, streamline the study of gene expression and protein interactions. Okay. Then another environmental and food testing. That is another biological applications of MEMS and NEMS, that is environmental and food testing. In that part, we can detect the, it is a bacteria, E. coli. It is just a bacteria. That is, we can detect contaminant of pathogens in food, water. Similarly, detection of pesticide residues. It is an apple. We can understand the various pesticide residues by using MEMS and NEMS technology. That is the another biological applications of MEMS and NEMS. Then there are many applications in advanced research. Okay, that is in this case you can see AI diagram, single cell analysis. So this, by using microfluidics, this isolate individual cells, which can isolate an individual cell for studying the responses to drug or stimuli. And it is a single cell analysis. We can isolate a single cell by using single cell analysis. In this case, that is a dynamic bioreactions. So we can continuously monitor the evolving of biological system, such as microbial culture or enzymatic reactions. So these are the two advanced research applications of biological applications of MEMS or NEMS: single cell analysis and dynamic bioreactions. Now we are just going to some questions. The first question: how does microfluidic channel works? Illustrated with a schematic diagram. So the answer will be the microfluidic channels are small scale passageways, typically 10 to 100 micrometer wide, which is designed to manipulate and transport tiny volume of fluid. So that is in nanoliter or picoliter range. So this channel form a core of microfluidic devices, which are widely used in biological, chemical, and medical applications. Now we are going to understand how this microfluidic channel works. So this operate based on fluid dynamics principles at the micro scale, where there will be laminar flow dominates. That is, the fluid flow in a smooth layers with a minimal mixing due to very low Reynolds numbers. Okay, Reynolds number, it is a part of some deals with the property of liquid. Okay. So how does microfluidic channel works? There will be laminar flow dominates. Similarly, there will be capillary action and surface tension. Then there will be precise control is achieved by micropump valve pressure gradients. So these three things. So first one, laminar flow dominates. Okay. The second thing, there will be capillary action and surface tension. Then there will be precise control, which is achieved by using micropump valve and pressure gradients. Then the working principle. In this figure, you can see there is a microfluidic device. There is a straight channel. It is a straight channel. It is a Y channel. Okay, it is represented another. Okay, so this is a microfluidic chip with the two devices. It is a first one straight channel strip. It is a Y channel strip. So we are going to understand how this is working principles. So the fluid samples are introduced via inlet, pipette or syringe into micro reservoirs. So there will be flow through channels, that is a pressure difference, electrokinetic force or capillary action. So these things which will drive through the micro channels. Then reaction or analysis. Along the channel, reagent may mix or reactions may occur, and which may detect the specific biomolecules. Then collection or output. Processor fluid exit through outlet, which is captured for further analysis. So there is a introduction, fluid introduction, and there will be flow through channels by pressure difference or electrokinetic force or capillary action. Then there is a reaction or analysis. Then there is output. This is 4 stage. First stage introduction, second stage flow through channels, third stage reaction or analysis, fourth stage collection or output. Then another question, describe the microfluidic process in solution flow system. So the answer will be, in a microfluidic solution flow system, fluid are manipulated and moved through tiny intricately designed channels to perform various tasks. So this microfluidic, that is which is placed on a chip. Okay. So this process involves precise control of fluid flow, mixing and separation, which allow for applications like chemical reactions, sample preparation, and biological analysis. Now we can understand the more detail. There will be microchannels and reservoirs. We are going to explain, we are going to describe a microfluidic process in a solution flow system. So microchannels and reservoirs. Then this diagram represents, you can see there is a microfluidic chip. There is this box that is given in the box, that is called a microfluidic chip. There is a computer here, gas pump here, regulatory module here, aqueous solution, lipid solution, and samples. Okay, this is the microfluidic chip. The experiment is running by using computer. There is a gas pump. Okay, that is a fluid injection and control. So which means that a fluid is loaded into device through inlet, and the motion can be controlled by using various methods. Some methods are involve pump, valve, external pressure. Okay, so we can utilize surface tension, centrifugal force, or electric or mechanical field to manipulate the fluid. Then mixing and separation. So once the fluid is flowing, they can be mixed uniformly in a micro scale chip. So which can be separated different substances with the solution, such as removing cancer cells from blood sample. Then these have a lot of applications, this microfluidic process. Chemical reactions, that is synthesizing nanoparticle. Then sample preparation for isolating and purifying viruses. Then they have a biological analysis like PCR or DNA analysis. Then there will be applications in drug delivery by using microneedles or other method. Then there will be flow cytometry. Then now last topic, we are going to understand the advantage of microfluidics. So there will be precision, there is a miniaturization, speed and efficiency, and cost reduction. Okay, these are all about MEMS and NEMS biological application in microfluidics. Okay, thank you.